Henning said, I won some sort of prize for travelling the furthest to this workshop, and that prize was I got to speak first. So thanks a lot, Henning. And in all seriousness, I do appreciate the invitation to come and speak here again, Henning. It's always appreciated, and to all the other organisers as well, Andreas, Tom, and everyone else involved. This is always a very valuable workshop, very rewarding for the speakers as well as the students, I think. And so I hope you all enjoy yourselves here and take a lot home out of the workshop. So the first talk that I'm giving today is on detergents and their use in, I'm going to be focusing on their use in the studies of membrane protein structural studies, with a major focus on 2D crystallisation, which is obviously what most of us do here, but also there'll be implications for studies of membrane proteins in terms of their function, in terms of 3D crystallography studies and things like that as well. And as Henning said, we should try and make this kind of like a discussion format as much as anything. So if anyone has any questions, feel free to interrupt, put your hand up if there's something you don't understand. Don't be shy and feel free to sing out. So by way of introduction to the topic of detergents today, I chose to introduce this through looking at membrane protein structure determination generally. And this is a slide that I put together around 2008, so it's a couple of years old now, but it still illustrates a fairly pertinent principle. And that is with regards to structural studies of membrane proteins as compared to the studies of soluble proteins. And what you can see from this slide here is that basically there's a lot of proteins that exist on the planet. Many of these are membrane proteins, depending on what you use to estimate what a membrane protein is, anywhere from 20%, some estimates put this as high as 1/3 of the global genome. And within the protein data bank, when I put this slide together, we had about 37,000 atomic models. Only 200 of these were membrane proteins. So despite the fact that about 30% of all proteins are membrane proteins, only about 0.5% of the structural information represented in the protein data bank represents membrane proteins. So these are grossly underrepresented. Several different techniques, by far and away the most successful is X-ray diffraction. However, if we expand this out to intermediate resolution studies, which of course is where some of the real power of electron crystallography lies, you can see that electron crystallography and electron microscopy techniques start to actually occupy more of the membrane protein structural space. So it is a significant technique in that regards. And just a quick update of some of these figures. So currently, I looked at this on Saturday, I think it was, and there's about 67,000 atomic models in the protein data bank now. So in the last couple of years, that's grown quite appreciably. About 670 of these are membrane protein structures, and what we call a unique membrane protein structure, there's about 200 of these. So this is 600 structures of which there's some overlap, but still indicative of the percentage that's represented there. So the take-home message from this slide is that the rate of membrane protein structure determination lags behind that of soluble proteins. So this is a plot that I took from a review article by Stephen White, and what he shows is going from year 0, when the first soluble protein structure was solved and also the first membrane protein structure, you can see the growth in structures relative to that first year. And even though they're both going up at a logarithmic rate, as an exponential rate there, membrane protein structures are lagging somewhat behind. So why is this a problem? Basically what this comes down to, in all likelihood, is the fact that membrane proteins are not happy outside of their native environment. There's a major difference between soluble proteins and membrane proteins. Membrane proteins live inside a hydrophobic environment for the most part, and when they're removed from that environment, they need to be protected. And this is what brings us to the topic of the talk today, and that's detergents. So a lot of these factors here which influence the ability to retrieve structural information on membrane proteins are related to that fact that when we take a membrane protein out of its native environment, it's not really happy. And we really need to optimise the conditions we use to retrieve our protein from its native environment. So one of the key concepts that ties into a lot of the things I'm going to talk about today is this concept of hydrophobicity and the effect that hydrophobicity has on the very ordered structure of bulk water. So bulk water, as shown on the left-hand side here, has a very ordered nature about its structure. There's a very strong, extensive network of hydrogen bonding, and when we introduce hydrophobic structural motifs into this water, it distracts that hydrogen bonding network and it results in a thermodynamically unfavourable outcome. And so then what the bulk water solvent drives is processes which seem to reduce the effect of that thermodynamic instability to a situation that the water is happy with, to a best thermodynamic principle. And so this hydrophobic effect, this restructuring of ordered water to accommodate a hydrophobic motif is the sort of thing that drives protein folding, in particular transmembrane protein folding as well, which has exposed hydrophobic regions. It drives detergent micelle formation, it drives the formation of lipid membranes and lipid bilayers and many other things. And as I just said, aggregation of hydrophobic surfaces offsets this thermodynamically unfavourable disruption of the water-hydrogen bonding network. So comparing soluble proteins to membrane proteins, what these two images show here are two proteins, one of which is soluble, one of which is a transmembrane protein. And the colouring on the surface here relates to the relative hydrophobicity of the exposed surface residues. The blue residues are relatively hydrophilic, and the orangey brown residues are relatively hydrophobic. And so it becomes quite obvious from looking at these two proteins that the one on the right here is a transmembrane protein. The orange regions, which go the whole way around the outside of the molecule, and the surface of this molecule is appreciably more hydrophobic than the hydrophilic soluble protein on the left here. These are the regions that within a cellular environment, within a biological membrane, these would be protected. And in fact, this hydrophobic region on the surface of the protein is something that allows the membrane protein to carry out its critical function. Lots of membrane proteins are things like channel gates. They gate the transport of ions and solutes across membranes. They may transduce signals across membranes in the case of things like G protein coupled receptors and things like this. And so it's not only useful that they're held within the membrane, but it's actually critical to their function that there's some way that we can hold these proteins within the membrane and lock them into this space. And this is achieved by this hydrophobic effect, this interaction of the exposed hydrophobic residues with the hydrophobic core of a lipid bilayer. Okay, so this brings us to amphiphils, which almost gets us to detergents. So amphiphils, there's a number of names that are substituted for amphiphils here. Sometimes they're referred to as amphipaths. Sometimes they're referred to as surfactants. Sometimes amphiphils are just referred to as lipids, and we delineate lipids as being single-chain lipids, double-chain lipids, three-chain lipids, and so on. By definition, an amphiphil is a molecule which possesses both hydrophilic and hydrophobic parts, or hydrophilic and lipophilic, as it's sometimes referred. So hydrophilic are water-loving sections, and lipophilic are lipid or oil-loving sections. And so this is illustrated in this diagram down the bottom here, where we have a typical amphiphilic molecule. We have a polar head group, which is hydrophilic, and we have a non-polar and apolar tail, which is hydrophobic. And amphiphils are important to the study of membrane proteins, because these amphiphils allow us to stabilise membrane protein structures and to retain membrane protein function once we take it away from this native bilayer environment. So when we think of amphiphils, there's probably three things that immediately come to mind: detergents, lipids, cell membranes, and fats or oils. And as a way of broadly classifying these things, we sometimes consider detergents as molecules which have a single hydrophobic chain with a hydrophilic head group, lipids as things which have a hydrophilic head and a double hydrophobic tail, and fats and oils as things which have three hydrophobic tails. However, these definitions are a little bit flexible. Sometimes we have things that look like this that behave more like this. As you can see, detergents are really defined as an amphiphil that's water-soluble. And indeed, short-chain lipids are often water-soluble because this hydrophobic region is appreciably small and can be shielded by the relatively large hydrophilic head group. Similarly, some detergent molecules behave somewhat like lipids, and we have some crossover here. Fats and oils are predominantly defined in biological senses as triglycerides. This is what we normally see within cellular organisms. But so these definitions are a rough guideline, but as I say, there's examples of compounds that break these rules and where compounds maybe fit slightly better into one of the other categories. So they shouldn't be considered hard and fast. However, what I'll be talking about mainly today in terms of detergents is in fact molecules which have a hydrophilic head group and have a single hydrophobic tail. So by way of an overview of what I want to talk about today, I've covered a couple of these topics already, the membrane proteins and amphiphils. I'm now going to go on to talk about detergents a little bit, the properties of detergents, classes of detergents which we commonly use in membrane protein structure studies. I'm going to focus on the uses of detergents in 2D crystallography studies, and I'm going to make some comments about working with detergents, and at the end some novel detergent-like molecules. If I've got some time, I'll mention those briefly. Okay, so there's a few different properties of detergent molecules, some of which you may have heard of before, some of which you may not be heard of before. Some of these are more important to structural studies than others, but either way, it's good to have an appreciation of what all these different these different properties are, and how they may be of value in terms of studying the structure of membrane proteins. So I'm going to go over these one by one, starting with critical micelle concentration, this HLB parameter, the critical micelle temperature and Kraft point, cloud point, and the aggregation number finally. Okay, so a micelle is a structure that a detergent forms to overcome this destruction of the H-bonding network of water. So this is, in most cases, the most thermodynamically efficient structure that water can force detergent into once detergent reaches a certain concentration. And that's the important thing here. These detergent monomers, which have a conical shape, usually because this hydrophilic head group is larger than the hydrophobic tail in terms of width, These are somewhat water soluble, but once a certain concentration is achieved, they tend to form these micellar type structures like the one shown on the bottom right hand side here. And this is something that's driven by that thermodynamic process of water disruption and trying to minimise the disruption of the water network. So this is the critical micelle concentration, the concentration above which we start to form these micellar type structures. And so as a bit of a guide to membrane protein structural studies, we usually use this critical micelle concentration to guide what concentrations of detergents we want to work with in stabilising and extracting proteins. For stabilising proteins out of a membrane, usually something like 2 to 3 times a CMC is something we would normally work at. When we're extracting proteins from a membrane, up to something like 10 times the CMC is where we'll work with. And this is largely dictated by the abundance of the protein within the membrane we're trying to extract from, and really it's the detergent to membrane lipid ratio which is important in determining what concentration we work with here, more so than the actual CMC of the detergent relevant to the protein. So this is a stylised cartoon view of a micellar detergent structure, and in recent years there's been some studies going on, and it's become more apparent that these micellar structures are relatively ideal, and what these things actually look like if we perform molecular dynamics simulations is tending towards these ellipsoid type structures. There's detergent monomers which are free in solution on the outside. We have these structures which aren't perfectly spherical, as that cartoon tends to suggest, and depending on properties like the aggregation number and the HLB of the protein of the lipid, of the detergent rather, we get changes in this ellipsoid structure from something that approaches a sphere to something that starts to look really elliptical, like this example here. And we have monomers that are in equilibrium with the micellar type structures, and these tend to sort of pop on and pop off like lipids do on cell membranes and so forth. So that's an example of what things really look like. So with regards to the critical micelle concentration, there's a few general properties and rules of thumb that we can draw from looking at critical micelle concentrations of detergents that are related to one another. So this graph on the top left-hand side here shows the trending of critical micelle concentration on the left. This is a log scale with the hydrophobic chain length of a couple of different types of detergents that are commonly used in 2D crystallisation. These are sugar-based detergents. Shown in blue is the multicide series of detergents, and shown in yellow is the glucoside series of detergents. And so basically what we've got here is the CMC plotted as we add extra methyl groups to the hydrophobic tails of those sugar-based detergents. The numbers used to construct that graph are shown down here, in blue for the multisides and in yellow for the glucosides. And by looking at plots like this and other similar plots of detergents which have related hydrophilic head groups and varying hydrophobic tails, there's a number of properties which we can draw on as general rules of thumb. As you can see in this diagram here, every time we add a methyl group, we get a decrease in the critical micelle concentration. So the longer that hydrophobic chain length is, the lower the critical micelle concentration tends to be. For ionic detergents, it tends to drop by about half every time we introduce a methyl group. For non-ionic and zwitterionic detergents, it tends to drop by about 80%, and hence we see something that approaches a log scale here. When we introduce branching into the hydrophobic tails, it lessens the effect by about half. Phenyl rings are equivalent to about 3.5 methyl groups. So aromatic hydrophobic regions have a different effect to aliphatic regions. And perfluorinated hydrophobic tails tend to have a lower CMC than the related hydrocarbon tails. If we do introduce things like saturations into these hydrophobic tails, we tend to increase the CMC. Cis has a greater effect than trans in the configuration there, and introducing these polar groups like oxygen or hydroxyl groups also tend to increase the CMC. Generally, CMC decreases when we start to introduce electrolytes into solution as well. So the CMC of a detergent in water probably won't be the same as the CMC of a detergent in the presence of a salt or an electrolyte, and that's to do with the effect that those counterions have on the interaction of the head groups associated with these detergents. Indeed, a lot of these properties here relate to the fact that the CMC is dictated largely by the interaction of these individual detergent monomers. And effectively what we see when we form a micellar detergent, a micellar type structure, is we've got these hydrophilic head groups which tend to be repulsive, especially in the charged detergent structures, and these hydrophobic tails which want to attract and aggregate together. And so the formation of a micelle is driven by the competing forces here, where the hydrophobic attraction overcomes the repulsion of the hydrophilic head group, particularly when there's a like charge there. So you can imagine that anything that increases the hydrophobic nature of this chain is going to lead to a tighter packing of the detergent micelle structure, and so this is then going to reduce the CMC. So this is a summary of some of those properties that I've just gone over, and also some others which I may not have mentioned yet. I may mention further on. And this is kind of just a reference slide, a quick reference slide, if you like. But basically, what it shows is if we consider the CMC, for example, it tends to be lower if we have a hydrocarbon chain which is longer. We get stronger binding to the detergent. The micelle packing tends to be tighter because we've got greater hydrophobic area in the tails. Membrane extraction is usually harsher. Dialysis is more difficult. I'll touch on that later because we have a low CMC concentration. It's difficult to remove. And working with low CMC detergents tends to be cheaper because we don't need to use as much of them, basically. It kind of depends on, so this is a general rule, but it kind of depends on the head groups as well, and it's to do with infiltration of the membrane. I can see Andreas is shaking his head, which worries me.
Well, I think the general rule is that the high CMC detergents, which are the short chain,
yep.
are harsh. They offer DOE or offer glucoside, they are pretty aggressive.
Yeah. Yeah, that's true, I guess. In terms of, I guess, so yeah. Yeah. So I guess what I'm thinking of here as well is things like SDS, which have a very, very low CMC, but that's related to the ionic head group. So so if we consider this strictly as a cross-reference of like head groups, then yeah, you're right. This would be back to front. Yeah. Okay. So thanks for pointing that out.
You can just change one or two letters and with a D instead, which means harder for the extraction. Can you go back one slide?
Yeah.
So you have a difference between membrane extraction power and a stabilizing effect on protein. So according to this, you need a high CMC to take it out of the membranes and then a low CMC to stabilize it.
Yes, because the high CMC is very harsh in extracting the protein, but at the same time has a very harsh effect on maybe stripping away extrinsically bound lipids or stripping away necessary cofactors. And so as I'll touch on later on in the talk, sometimes there's not one detergent which performs everything we need for purifying a protein. So a detergent which is very efficient at stripping a protein out of the membrane may not retain its function, and therefore we may need something that offsets the harshness of stripping that membrane protein out of its membrane, and something that retains its function and maintains it as stable. Does that kind of make sense? Yep. And so so some of these things I haven't really covered yet, and I'll talk about those a little bit later on, and maybe we'll come back to this slide towards the end of the talk. So that was CMC, which I mainly focused on so far. The next property I wanted to talk about is this hydrophile-lipophile balance, because what I've tried to do on that previous slide is relate a whole bunch of properties to increases and decreases in CMC. But what's actually become apparent from a few different studies now is that maybe the CMC is not really a good guide of the harshness or otherwise of a detergent, or the solubilizing power or otherwise of a detergent. And something that's a better guide is this property that's called the hydrophile-lipophile balance, or the HLB score of the detergent. And basically what this is it's a measure of the solubility of the detergent, if you like, the hydrophobic nature of the protein, and it tends to correlate fairly well with a number of properties in relation to using detergents to purify membrane proteins and also to maintain them as stable. So there's a couple of different equations which I've seen for calculating the HLB of a detergent. This is one of them. The second one is this. Now, I'm not sure. I haven't actually done enough calculations to see whether they give the same score or similar scores or not. But certainly this one here seems to crop up more frequently than the other. So maybe if you want to hedge your bets one way, maybe go with this one for now. But in any case, it's not critically important because most detergents have a stated HLB, which you can then work off. This normally ranges from 0 to 20, although for some detergents, the HLB value can be cited as being as high as 40. And the region of choice for most membrane-spanning detergents that we want to work with, for most detergents working with membrane-spanning proteins, I should say rather, is somewhere in the range of 12 to 20. For transmembrane proteins, usually something like 12 to 15 is a good HLB value for extracting the protein efficiently out of membranes and also retaining its function and its stability. Closer to numbers like 20 and 25 are the sort of detergents we tend to want to work with for looking at extrinsically bound proteins, proteins that aren't transmembrane proteins. So what this slide here will show is that if we try to correlate, this is an example of a G protein coupled receptor, which is published in Biophysical Journal. And if we try to correlate the activity of the purified protein with the critical micelle content of the detergent used, so you can see here the author that's carried out this work has done a substantial amount of detergent characterisation trials. They've come up with a number of measures of activity using a variety of different detergents. Each of these numbers here represents a different detergent. And you can see that there's no real obvious correlation between the critical micelle concentration of the detergent used and the function of the protein that's recovered. This score here, in terms of activity, will represent a combination of the yield of the purification and also the yield of functional protein. So low scores may indicate that we're not getting a lot of protein out and that it's functional, or that we're actually getting a lot of protein out but a lot of it's being killed. However, if we flip this around and plot it against the hydrophile-lipophile balance on the x-axis now rather than the critical micelle concentration, then what we find is that we start to get a reasonably nice trend. And it peaks in the middle here at a HLB score of about 14, maybe 14.5. And so this is about in that range I was talking about earlier, about 12 to 15 for working with transmembrane proteins. So it seems to indicate here, from this study at least, that this HLB parameter is a more reliable indication of a trend in terms of the ability to stabilise a membrane protein and the ability to extract it out of a membrane. And fishing back a bit further in the literature, we find that as far back as 1973, these studies here on, I think this was actually I can't remember what it was, so I won't say. But this is a transmembrane protein again, and again they've plotted HLB versus some form of activity here. It's a radioactive ligand binding assay. And you can see that once again we get this nice trend where there's a peak in the middle here at an optimal HLB score and a number of detergents which have a similar HLB which purify this protein relatively well. Yes?
Can you rationalize this in any way? How does the HLB relate to phototrophs or stabilizing function?
So it broadly correlates with the relative solubility of the detergent. So and I think what we see here, I've gone the wrong way. So let me get this right. HLB. So numbers up this end here have a large hydrophobic portion. Numbers down this end here have a small. No, that must be back to front. Small and large. So effectively, what you're seeing is I think a measure of complementarity of the relative hydrophobicity of the protein and the relative hydrophobicity of the detergent that you're working with. Did you want to say something?
Would a sort of simple-minded explanation be that if the hydrophobic tail is really quite much longer than the lipid side, then it's no good for it?
Yeah, so this HLB number is purely based on molecular weight.
Right.
So, yeah.
But what I'm trying to point out is those green, yellow, red, they fit quite nicely to the span of the hydrophobic belt around the membrane protein. And if you go to the right, depending, I don't know what the dispersion was, but the HLB so high would suggest that they have a longer tail.
Yeah, that may be the case.
And that might be a mismatch, which then creates difficulties for the membrane protein.
Yeah, I think that would definitely have something to do with it. I think in a broader sense, I think it's just a matter of matching up the hydrophobicity of the detergent with the hydrophobicity of the protein. Did you want to say something else, Tom? Or? Yep. I would agree with that view. I think that's right. Yeah, so I guess, I mean, you can see here that, for example, you've got C9 and C10 detergents, 48 and 49, which have a HLB that's similar to, for example, C11 and C12s, and they've got a different head group, different hydrophobic chains, but different head groups as well. So, yeah, I don't know whether anyone's ever looked at just purely looking at the length of the acyl chain, but I think well, clearly the hydrophilic head group has a function as well, because if you take ionic head groups, then they tend to be more destabilising. So maybe the HLB score reflects that as well in terms of the destabilising effect of the head group relative to the acyl chain length. But I'm not sure whether anyone's looked at that in isolation, and it may be worth looking at. Yeah.
The measured activity is actually the domain outside introduction mechanism, not actually the activity mechanism. So the whole plot here is actually the role of the decision is preventing the protein from aggregation. Otherwise, the CM, TPM probably counts for
yeah, this is radioactivity, so it's probably a filter binding assay or something like that. And so in this case here, it may not actually truly represent activity. It may just represent ligand binding, which is then inferred to represent activity. But yeah, I can't say for sure, but I believe this measure of functionality is representative of the protein outside of the membrane. Certainly the sort of studies that I've done in the past, that's how it's been quantitated. Okay, so those are probably the two parameters that are most relevant to studying membrane protein structure and function. And there's a few others that I'll just mention briefly now. The first of these is the cloud point of a detergent. So if you take a detergent solution and you heat it up, eventually you get to a point called the cloud point, where you get a clear detergent solution which starts to go turbid. And what this is thought to be a result of is the fact that as you start to heat this solution up, you get an increase in the number of micelles forming, and you start to get micellar aggregates that start to form. And these micellar aggregates are large, and they have the effect of refracting light so that the solution appears cloudy. And eventually what happens is you get partitioning of the detergent into a detergent-rich phase and a detergent-poor phase. And so it's been suggested by some people that cloud point may be of relevance for membrane protein studies, because what we can potentially achieve is a sort of purification procedure where we take a protein that has a low temperature cloud point, we solubilize all of the proteins out of a cell extract in this detergent, and then we heat it up beyond the cloud point, and what we get is partitioning of the detergent weak and the detergent rich phases. And the detergent-rich phase presumably contains the membrane proteins, the detergent-poor phase contains the soluble proteins, and we can partition our proteins in that way. The problem with this thinking, however, is that there's probably only a handful of detergents that have cloud points that are practical for working in this range. One of them is Triton X114, which has a cloud point of about, I think, 22 degrees. But aside from that, there's not many. So this is something that's been suggested in the literature that may have value, but whether it does in real life, I'm not sure how valuable it is. But regardless, I thought I'd make mention of it. This is a typical phase diagram of a detergent, and I mentioned the critical micelle temperature just now. Effectively, what this critical micelle temperature is a temperature above which we start to get micelles forming. So micelles will form above the critical micelle concentration, but only if the temperature is beyond the critical micelle temperature. Now, related to this critical micelle temperature, you can see in this phase diagram that this temperature being on the x-axis, this plot, where we have detergent micelles on the right-hand side, detergent monomers below the critical micelle concentration, and this crystalline state of detergents on the left here. Once we get above the CMC, we get micelles forming, but beyond the critical micelle temperature. And you can see that this Kraft point here is very close to the critical micelle temperature. The Kraft point represents a point in space where we're in equilibrium between detergent micelles, detergent crystals, and detergent monomers. But practically for most detergents, this Kraft point is actually pretty much the same as the critical micelle temperature. And finally, the last property I wanted to talk about is aggregation number. So the aggregation number is basically a measure of the number of detergent monomers within a micelle. So if we have 100 dodecyl multiside monomers forming a detergent micelle of DDM, then the aggregation number is 100. It's pretty simple. Not a lot of proteins have their aggregation number quantified, but for those that do, places like Anatrace and CalBiochem have the data freely available on their website. Most proteins that we work with for looking at membrane protein structure and function have an aggregation number of between 50 and 100, although there are some that are quite low, and I think CHAPS has an aggregation number of about 20. There's a couple of different useful calculations that we can carry out once we know what the aggregation number of a protein is. One of these is the concentration of micelles in solution. This is basically just obtained by taking the total detergent, there should be an equal sign in here, taking the total detergent concentration, subtracting the CMC and dividing by the aggregation number, and this gives us the concentration of micelles as opposed to the concentration of detergent. The micelle molecular weight can also be obtained from the aggregation number just by dividing by the monomer molecular weight. So sometimes that's of use as well, particularly if you're looking at doing stuff like size exclusion studies to try and purify away free detergent micelles from your bound protein or something like that. One other thing I wanted to mention quickly, and I won't go into too much detail on this because we're going to look at this in the practice. This is a detergent drop box which is located here in Basel, and basically this is a really neat way of quantifying detergents. I'm looking forward to seeing this in person. Most of the methods that are traditionally used for looking at detergent concentration are based on either dye binding assays or on some sort of effect on surface tension. And this particular instrument here makes use of the latter, the effect on surface tension of detergent. So if you've ever taken a detergent-rich solution of a purified protein or something like that, you'll see that the surface tension of that droplet is much lower than, say, an aqueous solution of pure water. And basically what this system does is it monitors that change in surface tension by looking at the collapse of a droplet, measuring something called the Young contact angle, which is the angle of this droplet. It describes the flatness of the droplet, and then by extrapolating this from a standard curve for that particular detergent, you can reasonably accurately measure the detergent concentration in solution. So this is something we'll look at in the pracs, and it's a pretty neat way of measuring detergent concentration accurately. Yes? I think I've asked that question the last 4 years. How many of us do you need to ask that question for it to be profitable, Andreas?
We went through that quite a few times. And the thing is, there are many little details about it which make it complicated. But, any workshop can make it, and the software, which is sort of the costly thing, is available.
Okay. Yeah, and there's a couple of points that I'll mention later on when we're talking about the ideal detergent for crystallisation studies, and one of the problems that many of you will have encountered, no doubt, is the fact that when you're concentrating samples which have detergent in them, you don't know what effect you're having on detergent concentration. So to be able to accurately quantify that can be a pretty handy thing. So, okay, so I'll move on. So that's a number of biophysical properties of detergents, which some are more important than others, perhaps, for looking at membrane protein structure and function studies. But that's a bit of an overview of the biophysical properties of detergents. So now I'm going to talk about the various classes of detergents which we usually work with. And so we can consider detergents broadly as being members of one of three classes. There's the ionic detergents, the non-ionic detergents, and the zwitterionic detergents. And as you'll see in a figure that I'm going to put up in a minute, detergents in this class here tend to predominate when we're looking at membrane protein structural studies. These are milder detergents. The head group has no charge, and this head group therefore has a low destabilising effect on the isolated membrane protein structure. Ionic detergents tend to be very harsh. These are things like SDS, as I'll show in the ensuing pages. Zwitterionic detergents are kind of a compromise between ionic and non-ionic detergents in that they have a net neutral charge on the head group, but because they've got some charge polarity there, they still have some of the nice features of ionic detergents retained. So cationic detergents are pretty hard to find outside of these quaternary ammonium-based detergents. CTAB might be the one that most people have heard of that's most common. This is the structure of CTAB down here. Anatrace in their catalogue only sell about 6 cationic detergents, and these are very harshly destabilising, harshly solubilising, and they're often not used in membrane protein structural studies. They're usually used in synthetic strategies where really harsh detergents are required. And you can see in this structure here, we've got the hydrophilic head group down here in this hydrophobic alkyl chain. these ionic detergents tend to be really good at disrupting protein-protein interactions rather than just disrupting protein-lipid interactions, and so this is why they're not really that well suited to membrane protein structural studies, because they start to rip apart important protein-protein contacts between, for example, transmembrane helices. Anionic detergents, for the most part, tend to be the same. Things like SDS and potassium laurate, as shown up on the top right-hand side here. There are a class of anionic detergents, though, which are based on the steroidal structure of bile acids and their derivatives. So this is the steroidal structure of bile acids here, and we have a head group shown up on the top right-hand side here. These detergents tend to form a really weird structure, so they don't have this characteristic hydrophilic head and hydrophobic tail. Instead they form this what's described as a bean-like structure, where you have a clear hydrophilic head group, but then in the tail you have a hydrophobic tail, but you have a hydrophilic face. And in this particular example here, if I remove the blue part, which represents hydrophilicity, you can see these hydroxyl side chains all poke out in the same direction. So the underside of this hydrophobic tail tends to be hydrophilic as well, and the top side tends to be hydrophobic. And so they don't form these neat micellar type structures, they form these elliptical bean-like structures. And for whatever reason, they tend to be less harsh in terms of solubilization as well. So compared to the other anionic detergents and the cationic detergents, these guys here are starting to approach the sort of detergents that we can implement successfully in membrane protein structural characterization. And indeed, a lot of non-ionic and zwitterionic detergents are now based on this type of steroidal type structure. So moving on to the non-ionic detergents, these are detergents which differ markedly from the ionic detergents in that the hydrophilic head group has no charge. There's two predominant classes, I guess, of these non-ionic detergents, the first of which is detergents which are based on polyoxyethylene glycol tails. Things like Triton X100, Tween, Bridge, all of these funny commercial names, as well as things like Octyl POE, the CNEN type detergents. These are all repeats of polyoxyethylene groups. And there's some examples of the structure shown on the right-hand side here. Triton is an aromatic based hydrophobic tail. Tween has this furan ring here, and the Bridge CNE8 Octyl POE type detergents have an alkyl chain as their hydrophobic tail. Okay, so the second major class of non-ionic detergents that we usually work with are those that are based on sugars, glycosidic detergents, if you like. So we have basic glycosidic detergents that are just based on the main sugar group. Things like glucosides and maltosides are the most common, as I showed the CMC plots earlier. This is obviously glucoside and this is a maltoside here. But also increasingly we have these modified types of sugar group-based detergents, things like thioglucosides, which are sulfur-linked. These cyclic maltosides, for example, which have cyclic hydrophobic tails, and also acid-linked sugars like this sucrose acid-linked sugar down here. And so these are used quite widely in studies of membrane protein structure, predominantly due to the fact that they're easily synthesized, they're relatively easy to obtain in high purity, and it's very easy to fine-tune their properties by, for example, carrying out some of these modifications like thioester linkages and so forth, which can have marked differences on their properties. So we can sort of fine-tune these detergents to a particular application that we want to use it for in studying membrane proteins. So they're quite popular, and I'm sure most of the people in the room are probably familiar with them and have used before. And so in between the ionic and the non-ionic detergents, there's zwitterionic detergents. And so these are detergents that, like non-ionic detergents, have a neutral charge in the head group. However, the zwitterionic detergents have a net neutral charge, so there's a positive charge offset by a negative charge. And so these offer, I guess, the combined properties of ionic and non-ionic detergents. They sit somewhere in between in terms of their harshness. And as I mentioned earlier, detergents like CHAPS and CHAPSO are steroidal-based detergents, which rather than having an ionic head group structure, they have this zwitterionic head group structure where you have a plus and a negative charge offsetting one another. And so these detergents offer some promise. Probably the most interesting application for these is the fact that they tend to have very high CMCs, and so we can use these as a stronger detergent so long as our protein is robust enough. We can use these as a stronger detergent to initially solubilize our protein away from the membrane effectively, and then remove this easily by dialysis, for example, or something like that. Also, detergents like digitonin, Big Chap, Deoxy Big Chap, which you may have heard of before, these are again related to the structure of these detergents in that they have this bile acid-like steroidal group, but they actually have non-ionic head groups in them. So you can see that even though those anionic structures which are poor on their own, the bile acid-based detergents for membrane protein structural characterization, they've actually laid the platform for the synthesis of these sort of detergents which are much more useful. The other major classes of zwitterionic detergents are these trademarked detergents, if you like, the ones that are named after the companies that produce them, I guess. Cal Biochem calls theirs Zwittergen, and Anatrace calls theirs Antigen. And this is a general structure of one of those. And again, you can see in these bile acid-based detergents, this bean-like structure predominates based on that steroidal hydrophobic group. And the final class of detergents, which is a subclass, I guess, of the zwitterionic detergents, are these lipid-light detergents. There's a few different examples now of lipid-light detergents. We have things like the phoscolines, which many of you may have heard of. These are detergents which a lot of proteins which previously weren't really happy in the standard sort of detergents you screen. You start to introduce phoscolines, which are lipid mimics in that they have a lipid head group but a single hydrophobic tail, and therefore they form micellar detergent-like structures but retain a lot of properties of lipid membranes, which are important for stabilising protein structures. These phoscolines tend to be really good for stabilising some detergents which previously people haven't had a lot of success with. The problem here is that they tend to be relatively expensive, and so working with them routinely can be a little bit cost prohibitive. Yes, Tom. Yeah, so I was just going to add that they tend to be weakly solubilising because they're so similar to the native detergent, the native membrane environment. They're based on a lipid. They tend to be weaker at extracting proteins out of membranes because they're so similar to the native membrane structure. And so well, there's definitely examples out there of proteins which have been successfully studied using lipid-like detergents. You were going to say something, Dan. You had your hand up.
Yeah, I know this interpretation, and maybe one explanation would be, from my point of view, is that the people who use phosphonine, they use to extract membrane protein expressed in eukaryotes, like in insect cells, and clearly the amount of protein that is solubilized is much higher with those type of detergent than with the others. But maybe an explanation is that it's also proteins that were present not at the membrane, at the plasmic membrane, but also in the other compartments, that are not fully maturated, maybe in the ear, and people do not purify the plasma membrane from such preparations. Do you think it would purify the plasma membrane so then you stop? Yeah. At least in our hand, we had such experience that we solubilized much more protein with phosphonine, and at the end, as you mentioned, part of the protein was not good enough. And when we purified the plasma membrane, we got a similar amount of purified protein with phosphonine and DDM in that case.
Okay, so that's phoscolines. They're one class of light detergents. There's also these lysolipids, which again are lipid-based single-chain structures. And as I mentioned right at the start of my talk, when I was saying about how the boundaries of what's a lipid and what's a detergent can be clouded at times, these short-chain lipids like DHPC, there's examples of these being used successfully to solubilise and crystallise membrane proteins in 2D as well. So that's the lipid-light detergents. Okay, so this is an overview, and again, this slide is a little bit old, but it's probably still representative. It's an overview of detergents used to either solubilise or crystallise membrane proteins and then obtain a 2D crystal structure of the protein of interest. And I've broken this up into ionic, zwitterionic and non-ionic detergents, and so you can see that the vast majority of successes, at least based on this data, are non-ionic detergents, although there are examples where some of these others have worked well. These examples down here of ionic detergents are those bile acid-based structures. And then I've also broken it up further into glycosides, for example, modified glycosides, polyoxyethylenes, lipid-light detergents, amine oxides, and the bile acid-light detergents. So you can see there's some quite nice trends there. And whether this is because sugar-based detergents are more commonly used, or whether it's because they're actually better at achieving results in 2D crystallisation, you can definitely see that the trend bias towards the use of these glycoside-type detergents in the success of 2D crystallisation studies. The blue is solubilisation and purification, and the red is crystallisation. So, for example, that you'd be familiar with, there's some examples where people exchange detergents or add an extra detergent in for the crystallisation, basically, and so that's why there's some slight variation in the numbers there. So I think this was taken from about 160 unique structures. So there's probably about another 40 out there which I could update this list with, but it's probably reasonably representative still. Okay, so now I'm just going to run through a few examples, a bit of data on typical approaches to working with detergents in terms of optimising your 2D crystallisation approaches. This is a non-ionic detergent screen that's been performed on a sugar transporter. And so what we've done here is we've basically used a number of different glucosides, maltosides, and deoxycholic acid to look at the solubility of our protein. So this is a pretty typical experiment that lots of people have probably done before. I'll just put it up for illustrative purposes. And this is really just screening the solubility of the protein, not the activity of the protein at this stage. So we've just gone through and screened at a nominal concentration the amount of protein that's present in the pellet and the amount of protein that's present in the supernatant after treatment with these detergents. And you can see that some are significantly better than others. In this particular example here, the 9-maltoside and 10-maltoside chains have almost entire solubilisation of the protein, whereas some of them have virtually no solubilisation and aren't appropriate at all. And so it's one thing to solubilise your protein out of the membrane and obtain a high yield, but quantity is often better than quality is often better than quantity. And so it's no good getting a lot of detergent if your protein at the end is rubbish. And this is a nice example here of a gated ion channel, which can be purified in Triton X100. It can be purified in other detergents as well. This is a typical trace here on the right-hand side where you apply negative pressure in a patch clamp study, and you get nice channel opening of one and then two channels here. What you can see here, when we purify with Triton X100, we get opening of two channels, and one of these channels is spontaneously opening, and when you remove negative pressure at the hash point shown here, this first channel continues to spontaneously open, which we shouldn't see. Yes, Tom? That's right, yeah. So this means even if you reconstitute it back into a nature's environment, you still have damaged the process to a degree that it does not recover even after reconstitution? It appears to be the case, yeah. So whether there's still Triton X100 hanging around and it's impossible to completely remove it, or whether it's actually done something to the protein and denatured it, the purification with Triton X100 in this stage obviously is not appropriate for further studies because it's had some sort of abnormal effect on the function of the protein. So when you do that reconstitution and test, would you do also the reconstitution of the pure lipid after solubilization with T800 to see whether the remaining triton would actually produce something? So whether, sorry, I'm not sure if I understand your question, but... So you pointed out that possibly there is also a normal hit because of the fact that not all of the T800 is out in your black lipid membrane. So this is actually reconstitution into liposome blisters. Okay. And then you patch those? Or what yeah, they're patched, yeah. Okay. But if you would do the same experiment, I don't know whether that is feasible, but just take lipids that are solubilized in T800, reconstitute the lipid vesicles, and then patch them. What would you do? Okay, so whether if we solubilise the lipids with detergent, so whether it's detergent hanging around with the protein or whether it's an effect that the lipid has on the detergent, yeah. That I haven't done that experiment, and that would be interesting to see actually. I mean, the easier thing is just to work with this detergent because it's better, but it's an interesting observation and one worth following up. Yeah.
So for the recrystallization, we normally look at how good is protein stability, whether the protein actually is stability, and also how nice is the shape in the gel-switching column. Is there any rationalization in this aspect that can correlate with what drastically focusing on and also
Are you talking about in relation to this work or just generally? Okay, maybe I can come back to that question at the end, because I think that's a bit more of a general question, yeah, if that's okay. Okay, so that's one example of a channel protein where Triton X100 clearly wasn't suitable, and this is another example here where this is a study of a G protein coupled receptor and a few different detergents that have been looked at for solubilization. This is work that wasn't done by me. This is work that's done by a colleague, and so basically what he found is that he gets the solubilization isn't great at all, but this is about as well as he can do at the moment. Unfortunately, he gets better solubilization in something like DDM or even SDS, and not surprisingly, SDS is detrimental to the function of the protein. But even DDM in this particular case here, if we look at our control membranes here, and then our DDM solubilized protein, the red bar is in the presence of inhibitor and the blue bar is in the absence of inhibitor. So basically when we take the inhibitor away, we should see an increase in activity, and this is a filter binding assay of a radioactively labelled ligand. And you can see it, even when we add back in lipids, which we think are important for function, we don't get any activity of the DDM purified protein. However, we do get radioactive label binding for the digitonin purified protein. So for whatever reason there, DDM's nasty to the protein, digitonin seems to be better. So the point I'm making is that it's important that your protein is functional, and it's not just a question of yield, solubilizing out of the membrane. So this brings us to some considerations of what we might look for in the ideal detergent, so to speak. And obviously, what I've already shown is that the protein must be stable in the detergent. It must be functional. We don't want it becoming denatured, and we want to preserve the native structure as far as possible. As I touched on earlier, if we need, during purification of our protein, if we need to concentrate the membrane protein solution and we have large micellar detergent structures, then these tend to become concentrated and don't pass through filters. And so a machine like Andreas's, which quantitates detergent concentration, is really helpful there, because we really can't guess what the effect of detergent concentration is when we start to concentrate our samples and so forth during purification. The possibility of detergent exchange during purification, as I showed earlier, some of the examples of proteins where they've changed detergents going from solubilization to crystallization. This isn't possible for all detergents, though. Some are more difficult to get rid of than others. So we need to consider this when choosing a detergent, and also the issue of purity. This is some old data from the lab that my boss used to work in England, Ben Hankemer, and so they were interested in studies of photosystem II structure. And what they found many years ago now, I think this was, about 10 years ago, they found that alpha and beta DDM have drastically different effects on the solubilizing power of proteins out of membranes. And this used to be a problem, because when you bought DDM from a detergent selling company, often they would just send it out to you and they would tell you it's high beta, but it wasn't always high beta. It might be 80% beta, it might be 99% beta, and there was no real quality control in place. Fortunately for all of us, this is not really the case anymore, and you can specify that you want less than 0.1% alpha, and you'll get less than 0.1% alpha in most cases. But certainly 10 years ago, this was a problem, and what this led to was, for example, with alpha DDM, we get these large supramolecular photosystem II complexes, and we can even isolate membrane arrays. With a similar concentration of beta DDM, we get nice homogeneous photosystem II dimers in this particular example here, and you can see there's one very small difference in the structure of these two detergent molecules, but it has a drastic effect on the properties of the detergent in terms of solubilization. And so it's important that you're working with pure detergents that are very well characterized, and that you can then predict reproducibility, the effects they're going to have in subsequent experiments. And so this brings me back to this example of our G protein coupled receptor, because it's been stated in some places, and I'm not entirely sure what the reason for this is, but I have some ideas. It's been stated that digitonin is a mild detergent, but it's unsuitable for crystallography. This was stated in a recent review in Methods in Molecular Biology. And I think part of this probably has to do with the fact that digitonin is a detergent that's isolated from native sources, and there's a bit of heterogeneity associated with digitonin preps. People that work with this detergent, which is often used in G protein coupled receptor studies, tend to, when they find a good batch of digitonin, they buy the whole lot and store it in the lab and use it for years and years, because getting a good batch of digitonin, so to speak, can be difficult, so I'm told. And so I think this probably ties in somewhat to the unsuitability of digitonin for crystallization, but there may be other reasons as well. So the previous slide that I showed about the ideal detergent really focused on solubilization. But in terms of purification and crystallization, there are some other things we need to consider as well. obviously the heterogeneity of the detergent can become an issue, whether it be to do with reproducible solubilization of the protein out of the membrane, or whether it's to do with, if we're looking at, for example, 3D crystallization in the presence of detergents, then all those detergents should ideally be the same. We don't want different detergent molecules in there. The detergent also needs to be easy to remove depending on what technique you're planning on using for reconstitution of your protein into 2D arrays. Some techniques are more efficient at removing detergents with different properties. Some tend to remove all detergents the same, and we'll see some talks on those later on today. And then the other thing to consider as well is if you're using a crystallization template type approach, not all detergents are compatible with all crystallization templates. So this is something else to consider as well. So this is the first slide I've got on 2D crystallization now, I guess. And so I'm sure you'll see these sort of slides a lot in today's talks and in the rest of the talks for the conference. But this is an example of the 2D crystallization procedure, which we can kind of consider as a reversible procedure. We take our membrane protein, we pull it out of its environment by introducing detergents, and we get these detergent micelle protected transmembrane proteins, and then what we want to do is basically take it back the other way, put it back into a native membrane under conditions which form 2D crystals. This kind of is a bit of a simplified view. Really what we've got going on here when we solubilise with our detergent is a whole bunch of different mixed lipid detergent micellar type structures like those shown on the left. And really what we want to do is not reversible, because when we go back, if we just go back to where we started from, we just end up with protein back in a membrane, and that doesn't really help us much at all. What we want to do is reconstitute into a limiting membrane under ideal conditions which lead to 2D array formation. And as I said, you may employ something like a template to try and achieve this, and if that's the case, then you need to consider the detergent that you're going to use. Just to briefly mention the different methods that can be used for removing detergent. So we have methods like microdialysis, biobeads, cyclodextrin. Dilution is another method, and these all have pros and cons. Dialysis, as a generalisation, has probably yielded the best crystals from electron crystallographic analysis. However, it can be difficult to remove, or near impossible to remove, low CMC detergents by dialysis. It can take a very, very long time. Something like dilution has had success where we dilute our membrane protein solution out past the CMC until we get reconstitution into the lipid membranes. However, you need a really high starting protein concentration for this approach, and it's not always practical to be diluting our sample out. And these absorption-based methods appear to offer some really convenient approaches to removing detergent. Most importantly, they tend to remove all detergents in a uniform manner. There are problems associated with biobeads, however, in terms of the heterogeneity of the size of these biobeads and the reproducibility of experiments. Cyclodextrins, which we'll hear more about later on today, I believe, appear to offer a nice solution to this problem. Okay, so in summary, these are the properties that I've just talked about in terms of what's ideal for our detergent for 2D crystallization studies, in terms of both solubilization, purification and crystallization. And you'll see there's some overlap in here. And I've already gone over all these points, so I won't go over it again. But what I will point out is that you need to keep in mind that one detergent may not satisfy all requirements, and therefore the possibility of detergent exchange or adding extra detergents in is one that we need to think about. And this is an example from someone that's in the audience today, I believe, where large sheets of this LH2 protein were obtained by introducing octyl glucoside into the crystallization mixture prior to performing crystallization trials. So this is a nice example, of which it's not the only example, where the detergents which help crystallization process may not necessarily be the detergents that you need to use in the early purification steps. Okay, so this is just an example of some of the detergent screening that we do in our lab. We routinely screen 5 different detergents, and we try to cover a wide variety of different functional groups in this initial screen. So we screen with DDM, OG, OTG, C8E5 and TX100, and these are some representative images from a preliminary 2D crystallization screen. And you can see that whilst I haven't indicated what detergent correlates with what image here, you get some quite drastic differences in liposome morphology. Yes, Tom? Yeah, the usual approach that we take is to optimise the detergent for purification and then try screening these detergents in the crystallisation procedure to see what effect they have. Sometimes we'll get prior information that, for example, one of these detergents is the protein's not happy in it. Like, for example, a protein might not like DDM when we do our initial solubilisation trials, and so therefore we tend to omit. We'd omit DDM from our crystallisation screening conditions and replace it with something else. It definitely makes a difference to liposome morphology. I have some results, but I'd rather not put them up on the screen here. But we can talk later if you like. Yeah. But yeah, it definitely has differences. I couldn't make a general comment about one being generally better than others. Like, for example, the thioglycoside from Mohammed's work, we don't see a general trend towards one particular detergent being better. And indeed, we've tried to screen detergents. They're all non-ionics, but they're not all non-ionics, or are they? Yeah, they are. But they're not all from the same class. They're all from different classes, subclasses. Okay, and so the last thing I'm just going to mention really briefly is some of these non-detergent surfactants. And I'm not going to go into too much detail on these because a lot of these non-detergent surfactants have been designed really for 3D crystallization studies. They're designed to stabilise the protein when you take it out of a native membrane. They're not really designed to be added in for solubilisation and removed again later on, which is what we're primarily concerned with for 2D crystallization. But just to illustrate a few design principles and the fact that this is a field that's growing and ongoing. So we have things like these amphipole type molecules which bind very tightly along their hydrophobic face, and then they have an exposed hydrophilic region. And the design principle behind these amphipoles is that the hydrophobic region binds to the hydrophobic transmembrane region of the protein, and we then have an exposed hydrophilic face, and these bind very tightly. They're held in place, and they're really good at preserving that native structure. Getting them back out again, though, is another issue entirely. Hemifluorinated surfactants are also something that have been experimented with, and these are interesting because they're not miscible with hydrocarbons, and so they therefore tend to be, at least theoretically, better at retaining protein-bound lipids. This is an example of a lipopeptide where we've got a helical peptide which has a hydrophobic alkyl chain covalently linked to the inside of the helical peptide. And like these amphipoles, you get this hydrophobic interaction with the transmembrane region of the protein and the helix forming a hydrophilic surface which maintains the protein is soluble. And a couple of other ones are these tripod amphipoles, which the overriding design principle of these is that the hydrophobic tails here are very rigid. And the idea is that if you introduce an amphipole which is more rigid, then you increase your chance of getting crystals because there's less flexibility in the amphipole, which is a critical part of a 3D crystal matrix, and therefore you're introducing more order at the start. You've got less things to order when you want to crystallise the protein. So as I said, a lot of these are designed for 3D crystallization and not so much for 2D crystallization. So they're worth mentioning, but I won't go into too much detail on them. And finally, probably the oldest of these, which started to get some attention back in the early 1990s, are these non-detergent sulfur betaines, which reportedly improve the yield of membrane proteins in solubilisation studies when they're used in conjunction. And it's been suggested that they have applications in preventing the aggregation of denatured proteins and maybe even in refolding of denatured proteins. Yes? Yeah, so one comment that I was tempted to make about these, but I didn't want to, is that you're right. Some of those ideas are a few years old. Some are 10, 15 years old, and they still seem to be boutique-like techniques, if you like. They're not widely used, and that alone suggests to me that they're more trouble than they're worth. And as I said, they're really designed for 3D crystallization, so to a lot of people in the room, they're maybe not of interest anyhow. But the fact that they haven't really taken off and they're not, maybe the non-detergent, the sulfur betaines are somewhat widely available, but most of the others aren't. And so I think that suggests that maybe they're limited in their applications. But that's certainly what I've found in searching the literature and so forth. Did you have a comment, Daniel? Or?
Yeah, concerning the amphipole, there are several extremely interesting applications. But regarding the crystallization, at least the 2D crystallization, I'm not sure that it's possible to remove the amphipole and replace lipids. Maybe, Catherine, you know much better.
Yeah, no, I think that's right, yeah.
So actually, you isolate the protein, you try to remove the detergent and you change to amphipole, but then after you work with protein in amphipole. For 3D, they got 3D crystals of bacterial reduction with amphipole. Yes, what I know.
But beyond that, there's not much more, right?
No.
Yeah, and so that suggests to me that maybe they're not that useful.
But
maybe there's not a lot of people trying them either. Yeah, no, this, so, yeah, if
The one that is interesting, which is the non-ionic transport, not available.
yeah.
The other one is a ionic, and the trouble is, of course, you can't get rid of them.
Yeah. But it's now available.
Yeah, but that one is also hot water.
Yeah, I mean, most of the examples of these things are a crystallization of bacteria rhodopsin, which has been known 1,000 times anyway. So, and so maybe it's reflective of the fact that that they're not widely used, as Andreas mentioned, or maybe it's reflective of the fact that they're just not more widely suitable. And certainly, from what I've seen in the literature, I'm maybe not qualified to answer that question. But yeah.
Do you have any comment on the fluorinated detergents?
On the fluorinated detergents? Yeah, so...
Are they detergents?
Yes, yeah, so the detergent-like molecules, they're not, they don't behave like lipids. So the
question is that I remember a publication in which actually they showed that it was not, proteins were not solubilized in microbes, but in emulsions. It was small emulsions of lipids, fluorinated detergent, and proteins.
Yeah, kind of like a small, yeah, sure, yeah.
But I'm not on you. You work with?
Structures like this are also a really hot area for drug delivery vesicles and things like that as well. So structures based on this sort of thing have been developed not just for protecting membrane proteins, but also for forming more rigid liposome-based drug delivery vesicles. And so I'm not sure how closely related these are to those sort of structures, but there's some interplay there as well. But yeah, I think the problem is they're not widely available, and so it's difficult to assess how effective they are, other than just to mention that people are doing this sort of thing, and that's about all I can say. Yes, Tom. Yeah. Mm-hmm. Yeah, and it's I mentioned it very briefly at the start when I was talking about the ideal detergent, that whether cofactors or extrinsically bound lipids are necessary for being retained through purification for function. I think Catherine's going to talk more about lipids as well, so maybe she'll mention it, the importance of extrinsic lipids. But yeah, you make a good point, Tom. Yes, I think we're nearly finished here. Yeah, so, well, the example that I showed there where the OTG was added in, OTG wasn't replacing the detergent that was in. I think it was a DDM, Andreas, that was in there initially? Mohammed, was it DDM initially that was in there? In your crystallisation studies where you add in the OTG to get the reconstitution, was it? LDAO, yeah, okay. So, and so that was the crystals were obtained by adding in OTG on top of the original detergent that was used. So there's definitely a precedent there for using mixtures. And I thought there's something else I was going to say, and it slipped my mind now. With regard to the HLB values, the hydrophile-lipophile balance, that is an additive property as well. And so it's been suggested from people that have studied HLB and in the pharmaceutical industry as well, where HLB is used quite widely for the solubilization of drugs and things like that. They often use mixtures of detergents, which they make up to have a specific HLB by combining, two parts of something that's got a HLB of 15 with 3 parts of something that's got a HLB of 18 to come up with a specific HLB in the middle, which is suited to their application. So in that regard, using mixtures of detergents to fine-tune that hydrophobic scale to suit your protein probably also has merit as well. Does that answer your question? Yeah? Yeah.
There was another group of detergent which was called the peptide protectors, which was developed and then...
Yeah, that's what... Kind of like that, yeah. Yeah, they're kind of like this. They're similar in that respect, in that they have again a hydrophobic face and a hydrophilic face, and they work on a similar design principle to the lipopeptides.
Okay, maybe we can. And keep in mind that the next topics are